To round out our three speakers before we get into a panel conversation among them and between them and you in the audience, we have Dr. Deborah Ashheim, who is an MD and on the faculty of the Health Policy and Medicine Department at Columbia University Mount Sinai School of Medicine. She's certified in cardiovascular disease and in internal medicine, board certified in those areas, and was specialized in heart disease and congestive heart failure. She's been published in a variety of prestigious medical journals, including the New England Journal of Medicine, and is going to talk to us now about the importance of clinical comparative effectiveness research to medical devices specifically.
Yes, so in fact, I think my comments are going to round this out a fair bit, because clearly I'm coming at this from a slightly different perspective. So a recent study published in the Journal of American Medical Association reviewed the scientific evidence that or the underpinnings of what are considered really quite rigorous clinical practice guidelines, the AHA-ACC guidelines. And what they found was only 11% of the guidelines were based actually on rigorous, high-quality evidence. So it's actually this particular evidence gap, frankly, as a clinician, as somebody who runs a clinical trial center, that for me and for those who do what I do, taking care of patients and trying to do it as best as possible, really drives the argument for really rigorous effectiveness research. But clearly the devil's in the details. And how is that research performed? When should it be performed along a continuum of clinical evaluation? That's really, those are the fundamental, really critical questions that have to be reviewed and carefully evaluated, particularly in the context of devices and device technology, which is really vastly different, obviously, than drugs. So as we all know, there's a very high degree of innovation, particularly in the US. If you look even just at 2008, there were 88 new drugs and biologicals that were approved. And in that same year, there were more than 3,000 new and modified devices approved by the FDA. And this doesn't even account for what was previously commented on, I think by both speakers, which are surgical procedures that are not related to products, but again, see an awful lot of innovation. Procedures such as living-related donor liver transplant. All of these pose drugs, the devices, and these surgical innovations pose clinical interventions that really require pretty complicated adoption decisions on the part of the clinician, as well as the policymakers, and frankly, the patients. So, in this context, there's clearly been and at this point I'm actually going to focus primarily on devices and more invasive devices, in fact, because I think it is a slightly more unusual dynamic that really is emerging in terms of the rigor with which it's being evaluated over the last couple of decades. But in that period of time, actually, there really has been this demand for increasing rigor with these types of devices in pre-marketing required for approval. But despite this rigor, there's still, frankly, are a fair bit of uncertainty that's left at the end of even a randomized trial for one of these devices. Effectiveness research, comparative effectiveness research as one pretty broad option really does pose a way of bridging this gap. But what are some of the causes of this uncertainty, particularly as we get very proud of ourselves creating these incredibly rigorous studies, and how might they actually influence when and how we do the effectiveness evaluations? So firstly, fundamental to devices, there are a number of unique aspects which actually drive some of these limitations. Most device evaluations are a comparison of a device to a device. You need to get your device on the market, and we all understand that. But, you know, in the absence of a comparator, like optimal medical therapy, optimal historic therapy, there frequently isn't really any evidence, primary evidence, supporting the clinical application of the intervention. Atrial fibrillation ablation is one example. There are a lot of catheters that are really good at ablating tissue. We know that. But in fact, we don't know if it's better to ablate the tissue than to treat somebody with medication if they have atrial fibrillation. But it's done a lot. Another critical consideration is incremental innovation. There are, as we all know, extensive modifications to the device itself and refinement of the procedures, both during the clinical evaluation phase and after marketing. And actually, this improves outcomes, but we don't necessarily evaluate it. We don't really know exactly what's going on. Operator skill is a distinguishing feature of devices, and clearly they impact outcomes and in fact the interpretability of clinical trials, as does other aspects of clinical trials, limited follow-up in terms of a time frame. Generally, if you're lucky, it's a year, and rarely are longer-term outcome measures studied. And again, sort of out of pragmatic necessity, trials are generally underpowered to look at secondary outcomes. However, those outcomes are frequently really the most valuable in terms of making clinical decisions. So just to step back for a minute, because there are a couple of these issues that I really think play a key role in terms of how you might want to consider implementing clinical effectiveness, comparative effectiveness research into the spectrum of evaluation, and when to do that. If you think about the impact of the operator's skill, obviously it's a fundamental issue. It actually, it primarily impacts the efficacy of invasive devices. You can't extract it from that efficacy outcome, the primary outcome. However, there's a learning curve. In the context of early development in a clinical trial, you're going to do it at a small number of centers, specialized centers. You will take steps to minimize heterogeneity in terms of the skill level of the doctors. Frequently use techniques like a run-in period, so the first handful of patients just don't count for your outcomes. Those are all great when you're really trying to create a meaningful, interpretable outcome for your trial. However, it does limit the generalizability of the results. What happens when it's not being done at 40 centers, but it's being done in 4,000 centers? And in fact, not surprisingly, both the safety and effectiveness of what was once studied really does change then once the technology is adopted.